All right, we're going to continue our exploration of Chapter 5 of Campbell's Biology, this time moving into the class of macromolecules called lipids. So concept 5.3, lipids are a diverse group of hydrophobic molecules. So lipids themselves is the one class of these macromolecules that are not true polymers. Okay, there are sometimes places where we can put things together, which is kind of polymer-ish, but it's not really made up of these individual units. Okay, so and some of these lipids do not even fit that category. So what's the unifying feature? Well, the unifying feature is the fact that they do not mix with water. They do not mix with water because they consist of mostly hydrocarbons, which are nonpolar covalent bonds. They can't hydrogen bond with the water, so the water forms a hydrogen or a water cage around it and pushes it out. Okay. The most important biological things that we're going to look at with these lipids are three groups: fats, phospholipids, and steroids. Now, fats are constructed from two types of smaller molecules, glycerol and fatty acids. Glycerol is very similar to carbohydrates, it's just lacking a carbonyl. But fatty acids have these long hydrocarbon chains, which are nonpolar in nature. We call it fatty acid because at the end we do have a carboxyl group. Carboxyl group can release a hydrogen and ionize, meaning that it is in fact an acid. How we form storage fats is we take three of these fatty acids and link them to a glycerol. We do this through a dehydration reaction. So we have a hydroxyl reacting with another hydroxyl. And in this case, we form what's called an ester linkage. It's called an ester linkage because it has an oxygen linking two carbons, but also has an adjacent carbonyl. We put three of these fatty acids onto this glycerol, and it forms what we call a triglyceride. So when we refer to a fat molecule, what we're really referring to is a triglyceride. These different fatty acid chains can vary in length, in size, and the number of double bonds that may be present in it. Now, when we start thinking about the inclusion of double bonds in these long hydrocarbon chains, remember that double bonds have a different geometry than single bonded carbons, right? So now we've moved away from the tetrahedral structure, which leads to this type of arrangement, and now we result in something that is more planar in nature. And this planar nature can form both cis and trans conformations. The way that nature uses is to put all of these in a cis conformation, meaning both chains are on the same side. Now, when you introduce this double bond, this actually creates a kink in the overall chain. This means that this can't pack quite as tightly, and it cannot form the same level of van der Waals interactions that saturated fatty acids can have. We call it saturated because it has the maximum number of hydrogens present. So it's saturated with hydrogens. Here this is unsaturated because it is not does not have the full complement of hydrogens because it has been replaced with a double bond. So the fact that these can pack tighter and can form lots of van der Waals interactions means that a triglyceride made of saturated fatty acid tends to be a solid at room temperature, but when you put even one unsaturated fatty acid in, it becomes more liquidy at room temperature. Now, you may have heard of trans fats. What trans fats are is simply an unsaturated fatty acid that has a trans conformation around the double bond. So instead of having both chains coming off the same side, they're off opposite sides. This is actually something that is not typically found in nature. And we started, this kind of came about in our belief that saturated fatty acids that are found in butter were unhealthy for you and would lead to heart disease. That is true, but in a more roundabout way. We can talk more about that in class. We know that things such as vegetable oil are much better for you because they have these double bonds. But we were interested in having something that could replace butter. So what we do is hydrogenate these vegetable oils, and this would artificially produce these trans fats that are in fact more solid in nature, and you can spread them like butter, but it turns out that these trans fats are even worse for you than saturated fatty acids. So sometimes science evolves, right? We were doing something that we think will help, and we were woefully wrong. Now, something to consider with these unsaturated fatty acids that we have the ability to make some of them, but we cannot make all of them. Okay, so there are in fact some unsaturated fatty acids that we cannot make. These we have to consume in our diet. Okay, we cannot live without them. One group of these is the omega-3, and also omega-6 are included in this. These are things that we need in order to produce everything we require in our body. As an added benefit, omega-3s tend to be protective against cardiovascular disease. And again, the reason for this is a little bit more difficult to explain. We'll maybe leave that for class. Now, one of the major functions, at least for triglycerides, is the fact that it is a good energy storage unit. So those long fatty acid chains, those hydrocarbons, can be broken down and turned into a lot of energy, and it is much more efficient than utilizing carbohydrate storage. Carbohydrates can be used quicker, but fats, you get more bang for your buck. Consequently, this is our long-term energy storage strategy. We have cells on our body called adipose cells. They are specialized to store triglycerides in little fat droplets, or little lipid droplets within the cell itself. This adipose tissue also serves other functions. It kind of cushions the vital organs in our gut, and it also acts as an insulator to our body. This is even more true when you think about things like whales or marine mammals that have very thick layers of adipose tissue to insulate them. So triglycerides are one group of fatty acids, or one group of lipids. The next group is something that we call phospholipids. Structurally, this is very related in that we have a central glycerol molecule that has two fatty acid chains. Now, instead of having a third fatty acid for the third carbon in glycerol, we instead attach a phosphate group, and then we can attach something else to the top. Here we have choline attached, but there are other options we can put up here. This produces a molecule that is both hydrophilic and hydrophobic, right? So there's one end that's hydrophobic, one end is hydrophilic. This leads to the ability to form something that is necessary for life, and that is something called a phospholipid bilayer. So if you look at our membranes, the things that separate the inside of a cell from the outside of a cell, it is these phospholipid bilayers. And you can see here that we have a hydrophobic head represented in gray, and then these two hydrophobic tails. The hydrophobic tails all point in the same direction on one face and then point in the opposite direction on the other face. And it allows us to produce these structures. We can take these and wrap them into spheres of varying different sizes, which leads to the compartmentalization within our cells. So these are very important, and we'll come back to the idea of phospholipids later on. We'll do a much deeper dive on membranes. The third group of lipids is steroids. Steroids do not seem to look at all like fatty acids, which you may think, well, how are we including in the same group? Well, again, it does tend to be mostly hydrophobic, so that is part of the linkage. But another part of this is this overall structure was actually formed from a longer hydrocarbon that had branches, and it folded in on itself, forming these rings. So in terms of origin, or in biochemistry pathways, they are very much related. The steroid structure produces a very flat plate-like thing, and it does tend to have both hydrophobic and hydrophilic characteristics. You can see its hydroxyl group at the end, which makes one lone point of hydrophilic interaction. We can take this core structure, which is called cholesterol, and then we can add other hydrophilic points onto it and form different things. And those different things can be hormones. So testosterone and estrogen are built off of this. We build several different things off of this core cholesterol structure. Now, it's also the case that high amounts of cholesterol in the blood seems to contribute to cardiovascular disease. Again, it's way more complicated than we think, so I'll leave that for class. Just to give you an idea of what this thing looks like, here's a structural image of it in 3 dimensions. We'll rotate it around. You can see all this green and white is going to be hydrophobic in nature, and this is the only lone little hydrophilic spot. Again, doesn't actually look like this. This is just the arrangement of atoms. If we actually look at it in more of a space-filling model, we can see that it forms a very rigid, tight structure. Again, mostly hydrophobic in nature. So, lipids are a diverse group of hydrophobic molecules. They are not true polymers. We can break them up into 3 larger groups: fats, phospholipids, and steroids. Fats have 3 fatty acid chains attached to a glycerol. They serve mostly as a fuel storage unit. These fatty acid chains produce a lot of energy. We can swap out the different fatty acid chains to those that are more or less unsaturated. That produces kinks. Those kinks lead to it being more liquidy in nature. We can also take this triglyceride structure, replace one of the fatty acids with a phospho group, and this produces the phospholipids. This is important for making biological membranes. And finally, our last group is the steroids, which are ring-like structures, but are in fact built from longer hydrocarbons that fold in on themselves, and like their cousins, is hydrophobic in nature.